what do we mean when we say n of 1? In n of 1, n is a sample size. It is a statistic that shows the sample size. So if I have a biomarker study where I'm looking at metabolites indicative of, like, let's say, endometrial cancer, and I say, okay, I have a n of 1,267. So that is my n. That is my sample size, right? An n of 1 study or single subject research, these represent really unique and highly specific forms of studying within the broader context of scientific research methodologies. So unlike more common broad scale studies, as I mentioned, that employ large groups of individuals and their goal is to deduce general trends or effects, the N of 1 study focuses on one subject intensively. And so this approach is designed to yield insights into an individual subject's response to interventions or behaviors or conditions that otherwise would not be visible in larger studies. So when we discuss N of 1 studies and how they fit into the broader scientific purpose, we should really, again, begin with a definition where it's a research design with one subject is observed and studied best over time, right? Usually over time. And so again, the N refers to the sample size, which in this case is 1. And the studies are particularly useful, Micah, for understanding the individual variability or testing the efficacy of treatments or interventions on a highly personalized level and exploring how phenomena that are otherwise difficult to study in larger groups yield themselves in an N of 1 design. So there's some features to this that are, very unique and desirable. So first is the aspect of personalization. So when we talk about personalized medicine or personalized therapies, we really have to conduct an N of 1 study on an individual, and they focus on this person, and the studies can be very tailored closely to the subject's specific conditions or their circumstances or their needs. So we employ what are known as repeated measures designs because we necessarily have to involve these repeated measurements and take these serial measurements over time to observe changes or responses to the interventions within that same individual. And in these scenarios, the studies use what are known as crossover designs. So basically, they're control methods that compare the effects of the intervention within the subject. So the subject essentially serves as their own control. And there are several advantages as well as disadvantages to such approaches. The advantages being, one, you get very, very detailed data. So these studies often provide an in-depth information about that subject's unique responses to interventions. They uncover nuances that, again, are missed by broader studies. And they're very flexible, which is another advantage. So they're adaptable. They can be adjusted quickly based on the study progresses and based on the subject responses. And as you can imagine, they're incredibly cost effective. So with only one subject, these studies are less expensive to conduct than larger, more arduous trials. And as I mentioned, there's the other side of the coin. There's disadvantages. Number one is generalizability. So if we want to figure out if, like, let's say a statin works for lowering cholesterol, there's better ways to lower your cholesterol, but let's say it's a statin. We want to make sure that it's generalizable. So the results of a single subject are not applicable to others. And therefore, it really limits the broader applicability of that study's findings. And there's no external validity, really. And then there's this complexity in analysis. So the statistics that are employed, right, you cannot do frequentist statistics with an N of 1 study. Frequentist statistics essentially fall apart, and the interpretation of the results are often complex, especially when trying to distinguish between the effects of interventions and what may be, I guess, in the context of metabolomics, what's very difficult is what are the effects of the natural interventions and what are the natural variations in the subject's condition. And there's, of course, ethical considerations too, because you're really taking, you have to really consider the subject's welfare and consent, because there is an intensive focus on one individual. And so usually what we see this in is three types of research. I would say three disciplines have really sort of led the vanguard in applying N of one studies. One is clinical research. Then behavioral science has been excellent in leading the charge in how to innovate these studies and apply them. And then there's educational research, which I don't really do, or I should say understand. And it's, they'll essentially use N of one studies to assess, let's say, the effectiveness of teaching strategies on a single student's learning outcomes, right? To figure out what's best for that student, rather than using like larger, schools of pedagogy or whatever. In clinical research, we're looking for instance, in testing the efficacy of any treatments for rare conditions where personalized medicine is in fact required, where it may not be feasible to get together 30 individuals, let's say, who have fiber dysplasia ossificans progressiva, which is a rare disorder of the soft tissue that ossifies to bone. That's not feasible. So we have to do an N of one study, or in behavioral studies, because there are very unique and highly specific behavioral patterns that are not susceptible to generalized study, and to tease those apart, N of one studies have been proven to be very important. And so, there's some things to consider when you're doing this. Number one, design. So making sure that the study is designed in such a way to ensure its validity, including the selection of appropriate.
Let's bring it back a little bit to kind of the broader concept of personalized medicine, because there's obviously a distinguishment between how it's used in science. Like, it is a scientific term, but now it's kind of being applied loosely to personalized medicine, which is just whatever that person is selling is essentially what they think it is, right? So what would you say, though? So we're taking this concept, N of one, right? So that's essentially the idea that people are using this buzzword. You just elaborated, well, it's a lot more than that. And so these are kind of some of the strictures that we use to ensure that we're doing an actual N of one study. But what would you say would be the hallmarks of personalized medicine in the future? Like, these are the things that really need to be in place for people to get good personalized medicine. And not necessarily in the context of a research study, but just in the context of personalized medicine, direct to consumer, direct to physician, that kind of thing.
Yeah. Gosh, that's a really good question. I'm not sure if I've ever really considered what are the hallmarks. All right. So when we talk about personalized medicine, and sometimes it's used interchangeably for precision medicine, and depending on who you ask, those are different. It's really transformative.
Start there. How would you distinguish those?
Personalized medicine usually has to have some component of genomic profiling, because that is the one thing that really personalizes it. But it's not enough, right? And if you only do genetics, then actually, like, you're missing out, right? So if personalized medicine really, and some people use it interchangeably, like I said, this is not a formalized like medical specialty yet. It's not. It's kind of fast and loose still. Personalized medicine in the theorists and the authors that I read and what we publish in the journals that are influential in this sphere, they do often make a distinguishment. And so personalized medicine has to have some component of genomic, and it can be a genomic component, ideally with downstream levels of omics also being integrated. But otherwise, we're really just getting into precision medicine.
So why does the genetic testing make the difference in it being personalized? I know you're getting into it deeper, but I just want to understand that.
Because when we say personalized, the one thing that makes it totally personalized to you is your genome, right? Like, that's the one thing that is personalized to you. Whereas if we were to take your metabolomic profile and my metabolomic profile and then change up our diets and our habits and everything for 6 months and then retake them, there's no guarantee that I could like match them up, right? That I could perform some sort of unsupervised clustering and then say, no, actually, this is, your sample is this one and my sample is this one. And this is, there's just no way, right? That's why, forensic analysts, when they find a specimen at a crime scene, they don't do metabolomics on a sample, right? They do genomics because it's truly more specific and therefore a much lower chance of incorrectly identifying or erroneously matching it to a certain person. So when we say personalized, there has to be usually some aspect of genomic medicine. But for the purposes of our discussion, we can just take them to be roughly the same. Okay, precision, personalized medicine. And so it's really this transformative approach. And the goal is to tailor that medical treatment to individual characteristics of each patient, whether it's the genome, whether it's the metabolome, right? And so the approach really considers differences in people's genes, their environments, even their lifestyles. And the goal is to just develop more targeted and therefore more effective interventions. And I've never considered what the hallmarks are. That's probably a topic of a good review paper.
I think that's
Yeah.
that's vitally important to the future of this, is defining out what are the staples of this that make it actually personalized medicine, because otherwise anybody can say they have personalized medicine.
Yeah,
But I mean,
we have like seminal papers like the hallmarks of aging, right? This is something that comes out every few years and kind of updates what we know on the process and the concept of aging. But I don't think there is one for this. Maybe there's a good topic for a review paper to write up. But
well, you're
really,
a pretty good visionary too. So even just like thoughts you have about it, as far as like, these are aspects that are really important. Maybe we're not getting down to the cornerstones on things yet, but I think it'd be interesting to hear some of the aspects that you think are pretty vital to that process.
So really it is taking it to a molecular level. So personalized medicine, has to use some sort of genetic or genomic or molecular data like metabolomics, proteomics, to understand the specific mechanisms that are underlying a patient's condition or disease. And if we can identify either genetic mutations that may underscore a predisposition to developing a disease, or if we can find biomarkers that are statistically related to that disease's pathogenesis, then healthcare providers can select treatments that are most likely to be effective for that particular patient. It is not just about figuring out, hey, what does this person have? Let's fit them into like a box of diagnosis and then treat that diagnosis. It really is this appreciation for the interconnectedness and vastness and the complexity of human biochemistry, and trying to correct that all the way from, again, the nuclear DNA down through the metabolites. So genetic and molecular profiling is, I would say, the number one hallmark. The next is, if the first hallmark is the tools that we employ, the next should be the outcomes we desire. And so I would say tailored therapies, right? So instead of just, we should customize this. So we do this through Ethereum. We do this through our digital twinning platform, right? So instead of giving these, generic recommendations, we can select specific drugs based on the patient's molecular makeup, specific formulations of supplements, customized treatment plans that account for that individual's response to certain medications, or even, in the virology space, the creation of personalized vaccines, personalized immunotherapies for conditions like cancer. And these have proven to be more efficacious than broad spectrum, sort of broad brushstrokes recommendations. When we, for instance, take a customer's data and we iterate, our database of interventions on them, and we may tell them to take niacin. It's not going to just be niacin. It's going to say something like, take, 250 milligrams of niacin in the morning with a fat-containing meal. Take another 250 milligrams at night to, there's a risk for you of experiencing flushing syndrome as a result of this. And so opt for an extended release formulation. And so that added detail, when to take the dosages, how to take the dosages, how to stagger them, whether to taper them up after a few weeks or taper them down. These are what our bioinformatics tools and the breadth and depth of our data collection enterprise now allows. It is no longer about telling someone to take niacin, read the back of the bottle. Sometimes that may be the case, but 98% of the time, it is a very specific and tailored dosage that this individual needs that we can assess from the metabolomic profile. That goes to the next hallmark, I think, is risk assessment. And when you can assess risk, you are preventing disease. And if you're preventing disease, then we're starting to, again, talk about that medicine 3.0, that preventive, proactive rather than reactive model of medicine. So if we can analyze in the context, let's say, of our Aristotle test, an individual's metabolomic and lifestyle intervention, then that personalized medical approach can provide our customers with an assessment for their risks of certain diseases. This invariably enables early intervention strategies, lifestyle changes, and monitoring to prevent diseases from developing or catch them at a more early, more treatable stage where that pathogenic process is most likely more tractable.
This is kind of interesting, and it'll be interesting as it develops out because there's a number of things that you can kind of outline in terms of, hey, personalized medicine requires genetic testing. Personalized medicine requires tailored interventions. Personalized medicine requires like a standard of success, right? But I think right now, as it currently is these are fairly loose definitions that a lot of people can meet. So I think the next level in this, right, is that we're going to be setting some sort of standard of like, well, this is the gold standard. This is the benchmark in which you have to exceed to say you're truly doing personalized medicine. Otherwise, you're just kind of a wash. Like, so it'll be interesting to see if like that's something that we can kind of set forth, right? Is this standard of personalized medicine in terms of outcomes? And that's what Chris was talking about too. When we were at dinner with him, he was talking about, well, you want to get for insurance reimbursement. It was in that context, but you want to be able to show that you're reducing the amount of hospitalizations or you're improving the outcomes in these people so that you can get insurance reimbursement. But I think in this context, it's kind of interesting. Like, how would we get to the point that we can potentially be setting a standard to this?
Yeah, that's going to come with many years of development, both in the science and then also the logistical improvements. And so this is really something that there are economic and ethical considerations surrounding this. So there are, right now, the tools that we use to collect the kinds of data we need in order to personalize treatment approaches, in order to make our interventions more precise, precision medicine. These are very expensive tests, right? Like compared to traditional, laboratory diagnostics that are currently being used with, ELISAs, immunofluorescent, et cetera. What we are using is a much more expensive, costly platform, and that cost in a business model is incurred by the customer. And as we develop, and not we personally at Ethereum because we don't have a hand in this, but as the engineering side begins to develop more cost-effective testing platforms, as we start to be able to multiplex our assays such that we're able to capture more with less, then we will, I think, reach a cost-effective tipping point and make it accessible to all patients. Right now, we haven't formalized a way to go about our data collection efforts and to get together enough research on a single, let's say, assay, on a single testing platform that can be leveraged essentially to get insurance reimbursement and convince, I guess, the actuarial side of health testing that this is indeed a benefit. And it's clear to every scientist and physician working in the field that proactive medicine costs far, far less than reactive medicine. And we know this. This is not a major claim. And we've seen this in studies. We've seen this in commentaries and opinion papers. There is everything from colloquial evidence to hard empirical science to prove it. But it needs to be sort of assay specific. And we're not there yet, where somebody has come forward and says, we've tracked our cohort for 10 years and we have reduced hospital admissions by this much. We've reduced medical claims by this much, and this is how much our test is costing, and this is the cost of follow-up treatments as indicated by our test. That is really something that still needs to be done. We understand that generally speaking, personalized medicine is more cost effective, but that's not enough to convince the insurance companies who are already making a lot of money and aren't necessarily incentivized to switch their models and switch the paradigm just yet. There's a lot to go along, right? Because it's not just regulatory approval for these treatments either. When you go through the normal regulatory process to get a personalized treatment, right? We did all these testing, now we came up with a personalized treatment. So does that mean that treatment, since it's new, needs to go through its own regulatory approval, even if it was personalized for just one person, right? So when we do frequentist statistics, when we do generalized studies, we have a randomized control trial, you do that one trial, you generalize it, and you show statistically it's applicable to more than one person, and you go through the approval process for that treatment, right? Thereby, it is cost effective in the long run for pharmaceutical companies. But if you create a customized drug, go through their proteome, go through their genome, and you identify drug targets and potential agonists that can be developed and synthesized in the lab, you then go and synthesize those, and then you want to give it to this individual. Does that mean that one personalized treatment then has to go through its own approval process to then be given to just one person? That's not currently cost effective. There's also
it'd be the methodology that's assessed, not necessarily like we would essentially need a new thought process that's used.
Right. We need new laws.
Yeah.
We need new laws for this. Right now, the law says no. Every time there is a new formulation, this has to be approved again, and that is just not. That's too high of a barrier for people to really start developing targeted therapies. There's also privacy considerations. If it's not for the litigation, the legal restrictions that need to change, there are privacy protections handling genetic data, which is of critical concern. And there's also ethical guidelines for how that personalized data is used in care. So until we line up the regulatory and ethical pieces of this puzzle, we are not going to get to the economic considerations, which will ultimately make these tests and these approaches more accessible.
So as far as technology for doing targeted therapies based on metabolism, whatever, is it always genetics as of now is what I hear most often, but you could do it on the metabolism too, I would think.
You should in fact do it on the metabolism, because when we were discussing this on an earlier webinar this week, if you have a genetic mutation in something, you have a variant. First of all, there's no guarantee that variant is making it to translation to RNA, or that RNA is then being transcribed to RNA and then translated to proteins. There's no guarantee. What we are seeing in the metabolome is necessarily you have some genes for these things that are then being expressed to RNA, then being folded into proteins appropriately, and they have the appropriate form, they carry out their function, they interact with your gut microbiome, and we are seeing the downstream proof of that expression. We are not looking at what could be, as in with genomics, but we're looking at what currently is. And so metabolomic data, again, for most of the conditions that are the greatest killers of humanity and account for the most loss of human life per annum, when we look at those conditions, they're all metabolic conditions, right? They're not genetic conditions. True that things like Alzheimer's and cancers, they have genetic components, right? Your expression of certain genes will determine if you have triple negative breast cancer or if you don't. If you have the APOE4 allele, APOE4, you're going to have most likely a higher risk, much significantly higher risk of early onset Alzheimer's. And that's what we see with familial inheritance patterns of Alzheimer's is this 4-4 allele. So, of course, it's going to have some influence, but that influence doesn't allow us to adequately assess risk and look at what's happening currently. Where are you at in the Alzheimer's pathology? Do you have any indications of underlying, tau, tangles, and plaque burden? Do you have, all of this pathology, but you have no symptomology yet? Do you have the pathology and symptomology, but the symptomology is relatively consistent with, age-related declines in cognitive function, which are termed mild cognitive impairment? Or do you have Alzheimer's, right? And so currently, Alzheimer's can really only be diagnosed. The gold standard is postmortem autopsy. And so where are you in that timeline? We can test that with the metabolism. We cannot test that with genetics, right? All we know is that you have a greater risk of developing it at an earlier age, or maybe for breast cancer, you are at greater risk of being non-responsive to hormone therapies. That is what we can tell with metabolomics. So it definitely makes most sense to do that. Ideally, you would integrate multiple types, right? Ideally, you would have genomic, proteomic, metabolomic, and some clinical data to go along with it, and you could gain a very comprehensive understanding of this person's health status. And this holistic view would definitely enable far more accurate diagnosis, prognosis, treatment strategies that can be used as a barometer for that individual's response to treatment. And a very important thing is that in the era of AI and machine learning, which, machine learning has been around forever. AI is a little bit more complex type of machine learning. But, I've been using simple machine learning in my publications since my early graduate school days. So things like random forests and support vector machines, these are now kind of looked at as a little bit more simplistic models, but they are able to analyze vast amounts of data, the kinds of data we need for personalized medicine from individual patients, and we can identify patterns and predict outcomes and personalize, again, those treatment plans. And these technologies and these approaches are crucial once we sit down to try to make sense of this complex, multidimensional data that's necessary in personalized medicine.
I think personalized medicine is the future. I very much believe that. But I also wonder, so you and I, we're actually more similar than dissimilar, right? We both need an adequate amount of sleep, be that 7 to 9 hours. We both need a diet of similar macronutrients, maybe some variance there. We both need to exercise on a regular basis. I think in the context of people who are sick, it's blatantly obvious, but I would love to hear from the horse's mouth. Like, do you think there's a point that we will be over analyzing people, that we're looking at the individual too much when there's so much redundancy inherent in people that we can make justifications for their health just by doing that?
Yeah, absolutely. So when we talk about redundancy, there's this redundancy in biology. And so if you have, let's say, the TCA cycle, we look at 5 of the 8 metabolic products of the TCA cycle. And if we look, and we also look at the cofactors, right? So we're looking at, niacin and folate, and we're looking at things like glycine and serine and various amino acids like glutamate, glutamine, alpha-ketoglutarate, proline, arginine, all of the amino acids that plug into the TCA cycle via anapleretic pathways. If we, for instance, now added on a 6th TCA cycle metabolite, we wouldn't actually get much more insight because we've already mapped it so densely that adding on a 6th metabolite isn't really worthwhile and wouldn't show us much more than we could just map our current panel to the pathway and infer. So we can infer if there is a reduction in the number or activity of an enzyme, or we can infer if the absence or the presence of a cofactor is facilitating or diminishing the rate of an enzyme's reaction. So we can do those things, and we don't necessarily need to add a 5th or a sixth outcome, right? And then also there is this law of diminishing returns, really, when it comes to AI algorithms. These algorithms are becoming more sophisticated on a daily basis, daily. And therefore, the value of data will start to become less, actually. So in the future, we may not have to go out and do 126 metabolites, a whole genome sequence, 900 proteins, 600 gut bacteria, and we will be able to identify more efficiently the salient features that enable powerful predictions, and therefore collect a smaller set of data that still has the same analytical predictive capability and explanatory capability than going out and collecting these large swaths of data, which is currently the norm in personalized medicine. So as these algorithms become more advanced, as these AIs become more advanced, the value of the data that we collect and the value of each additional variable we add to our collection enterprise is less and less and less. And then that's something to keep in mind as well.
Yeah, and then that kind of begs the question too. So there's other data points as well, such as wearable data, metadata, what you're eating on a daily basis, all that kind of stuff. Well, we have fed this engine of AI enough information to where that's integrated. Like, so we've mapped out the human biology more or less, and we've done it with 50,000 people, multi-omic integration. Like, we have all their layers of systems biology. At that point, do you think AI will be able to just make inferences on what it is just based off of metadata, such as what they're eating, their activity level, their sleep, and those various things?
Yeah, I don't know if we'll be able to get to the point where we can, infer underlying biology from just the metadata, but it will get to a point where with a few metadata points and a few key genes and a few key metabolites and a few key proteins and a few relevant bacteria, or maybe we just get your diversity index, your alpha diversity, and we look at your gut diversity, and we get these sorts of maybe more gross measures. We'll be able to tell the same things that we can currently tell with very large, advanced, AIs that are equipped to handle chemometric data, like our own AI. So that is something to keep in mind. And you're right, we will get to the point where it will be just a few easy tests, looking at less variables than we currently need to, and we'll still be able to probe just as deeply. But that comes with time. That comes with assessing both, like Bayesian frameworks, as well as like this frequentist framework, as well as, integrating the AI pattern recognition capabilities that are out there. We will get to that point for sure, Micah. Yeah. Yeah. I mean, we really want to talk about a type of statistics called Bayesian statistics that I've kind of alluded to. Really quickly, I want to go through what it is and what it's not. So when we think of generalized studies, we are thinking of, I thought there was someone behind me. When we're thinking of generalized studies, we're thinking of—
It was Bayes.
Thomas Bayes, is that you? No. And so if we are thinking of generalized studies, what we do is we put 30 people into a control group, 30 people into a treatment group, and then we do like significance testing based on their distributions, and we're trying to find significant features, and we're really moving away from this model of significance. And it's not possible with N of 1 studies. A Bayesian analysis offers this really powerful and really flexible framework for interpreting and analyzing this data in the N of 1 studies, especially when we talk about things like metabolomics research. The whole concept of Bayesian analysis. And the formula is essentially the probability of A given B equals the probability of B given A times the probability of A divided by the probability of B, right? So it's a very simple, but the elegance is actually in its simplicity, but it's very flexible, and we are integrating prior knowledge with current data to update our beliefs or our hypotheses in the light of whatever new evidence is at hand. So it's really well suited to the sort of personalized and iterative nature of these N of 1 studies, where the prior information about an individual's health about their metabolic state, response to interventions, can significantly inform the analysis. And I mentioned this in the context of an individual we tested who, all we can tell from his metabolomic data was that he had heavy metal toxicity. But what we could tell from his survey data was that he was a retired dentist. That tells us, right, it shifts the probability of the model and says, it's mercury. This person was most likely handling dental amalgams. It's mercury. So that's how we incorporate prior knowledge to tease apart really complex relationships that otherwise would be non-specific. If we didn't have that metadata, all we could tell as individuals is that you have some form of heavy metal toxicity. So Bayesian methods, they start with prior probabilities, which represent the existing knowledge we have or beliefs about the world, parameters being studied before any new data is collected. And so in the context of an N of 1, this could include, again, information about the individual's baseline metabolomic profile, the expected effects of dietary interventions based on previous studies, or just general knowledge about metabolic pathways, right? And so as new metabolomics data is collected on this individual over the course of the study, the Bayesian analysis updates its prior probabilities and it forms what are known as posterior probabilities. And so this process really involves calculating the likelihood of observing that new data given the different parameter values, so the level of a specific metabolite, and then combining this likelihood with the prior probability to really update our understanding. And it iterates, and this process allows for this nuanced understanding that reflects both the individual's data and then both the broader scientific knowledge. So this really is for personalization, for handling uncertainty, for flexibility, predictive power. These are all very much baked into a Bayesian study. And so we do various things in metabolomics with Bayesian data. We can do hierarchical modeling. So Bayesian methods can handle this multi-level data structures. So let's say we have metabolites nested within metabolic pathways. This Bayesian approach allows for comprehensive analysis that considers the dependencies between metabolites, especially as we consider, again, the redundancy between biology. And then for this longitudinal data, which is necessary in N of 1 studies, it provides these dynamic models. So we can, in metabolomics, we rely on what are known as Bayesian state space models. And they capture time-varying nature of metabolomic responses. They provide these deep insights into how an individual's metabolic profile can evolve over time. And then we can integrate that with other omics. So we can integrate it with genomics, proteomics, et cetera. And we allow for a systems biology perspective that considers these multiple biological layers, each one's posterior probability being affected or triggered by the prior probability of the other. And so this is how we can get around the sort of inheritively restrictive nature of an N of 1 study, because we don't have a comparison group. We don't have somebody else to sort of compare to. We can't do frequentist statistics. Who are we comparing it to, right? As I mentioned, they often serve as their own controls, especially if you can implement like a crossover design. But ultimately, this integration of multi-omics will require, we feel at Therion, a Bayesian framework, and that's what we do is Bayesian machine learning or Bayesian AI algorithm that we employ.
Well, to relate it back to kind of human nature and maybe in more simple terms in the context of health testing, but like if I were to tell you, Paniz, that, hey, you have heavy metal toxicity, the first thing that would come to your mind is not, oh, how did that guy over there get heavy metal toxicity? The first thing you would think about is, how did I get heavy metal toxicity, right? You would be thinking through, I had exposure to this. And that's kind of what Bayesian statistics is doing in a sense, except with a much more objective look at it. And it's saying, hey, your homocysteine level is such and such. And like, you wouldn't necessarily equate that to like, some sort of toxicity, but Bayesian statistics are able to do that. And so it just follows that logical flow chart, but does it in a very objective, scientific, and mathematically sound way. Whereas like the other form of statistics that you mentioned would be like, hey, Paniz, you have heavy metal toxicity. And you'd be looking around and saying like, how did all those people get heavy metal toxicity? And then trying to relate that back to you.
Yeah, and Bayesian statistics, when you look at like the scientific advancement of statistics as a discipline, we've always known that this was the answer. Like we have known since 50s, 60s, 70s that this was the correct way to do statistics, because there's a fundamental difference in the philosophy that one assumes when doing Bayesian statistics or when one does frequentist statistics. The problem wasn't that statisticians differed as to which camp they should go into. The problem was we didn't have the software and the computational ability to do Bayesian statistics. And so really there was actually a slew of papers between important statisticians in the 50s up through the late 70s, early 80s, that really were going back and forth and saying, okay, is Bayesian better or is frequentist statistics better? And everybody agreed that Bayesian was better, but they ultimately relied, came across the thought that it was like a big fat so what, because we can't do Bayesian statistics, right? The computation, the calculation by hand, especially with the computational abilities that were available at the time, it was an untenable ask. And so now that we have finally caught up, there has been this incredible revival and this renaissance of interest in Bayesian statistics and what it means and how it differs in philosophy and what it can show us. Yeah.
Well, I think that's a good point to end on. How do you feel?
Yeah, I'm great there. I just wanted to get that little bit about Bayesian stats out of the way because it is integral to what we do at Therium. It's the backbone of our analyses, really.
Yeah. It's so cool because there's so many cards that we can pull out at any given moment and be like, hey, look at this cool thing we're doing, between the Bayesian, the blockchain, the digital twinning, et cetera. And I know they all collaborate together, but it is pretty cool. Just all the different things that you and Alex have envisioned and are bringing into reality.
Yeah, we're tinkering with it. So hopefully in the next, 6 months to
you're not a candle maker. You're not tinkering with it.
No, I feel like I'm a very bespoke toy maker. You know what I mean? Like I make toys for little rich kids, and that's great. We have a lot of fun, and it's great to see that we have this business that tests people and gives insights into their health and is able to really zero in on a lot of conditions and help people make a vertical advancement in their well-being. But also we're scientists, right? We conduct studies, we publish these studies. We don't really, a lot of people want to do studies to show an insurance company or publish a white paper on a preprint server and sell their product. We do studies to publish them and get grants and sort of really move the needle forward in this space of digital twinning. And so if anyone's actually reading this or seeing this and wants to contribute, I am guest editing a metabolomics and a special issue of a journal known as Biomedicines. It's a great journal, wide readability, impact factor 4.7. So if you are a researcher seeing this and you want to submit anything that's within the scope of the special issue is known as Metabolomics and Bioinformatics Technology for Personalized Medicine. So if you are doing research and you want to submit a paper, please do so. I am in need of 2 more submissions, and the deadline is April 30th. And so please find me. You can either email my institutional email address at ASU or find my Therium email address and submit your work. But we're really fortunate that we have the business aspect, but we are also doing lots of great research on interventions, on biomarkers, on algorithmic methods, and we kind of have our hands in all of these pots. But hopefully it'll not just be for naught, and we'll apply these to individuals and really move the needle on their health.